I too want to welcome you to this first Fall Summit. We are very excited about the program. Dr. Lucy Bruin and Dr. Michael Benatar are here to share the latest information with all of you, to answer questions, and then there will be an opportunity this afternoon for you to tell your stories and to interact with one another. We so appreciate you making the trip here and to be part of this, because without those of you in this audience, we can't do the research. Dr. Benatar can't do the research. Dr. Bruin can't use the ALSA dollars that go out to research to help uncover the mystery that still remains around ALS. And certainly it is our goal as the association to bring all of the powers together, all the powers that be, to find a treatment for this disease and ultimately to find the cure for it, and most certainly the cause. So on behalf of the association, I welcome you here. We're delighted to have you. I hope you enjoy the next 2.5 days. And of course, the culmination will be the opportunity on Capitol Hill to influence your constituents who really control the dollars that come from the federal government. So again, thank you for participating. We appreciate it and hope you get as much out of this as we're anticipating that you will. Thank you. And I forgot to introduce Dr. Lucy Bruin, who is here with us. She is the Chief Scientist of the ALS Association. Welcome, Dr. Bruin, and she's going to kick off this program.
Welcome to all of you, and so pleased to have you all here and very excited about the program. Before I start my brief commentary to sort of give a context to the upcoming presentations, I did want to thank Michael and his team for their hard work at really putting the program together and being here today. And in fact, you'll hear more about their research, but it's really crucial, the kinds of studies that they are doing, and we're so pleased that we can support some of them. So just to put a context on it, one might say, so what do we do in our research programs for familial ALS research? And actually this is a tremendously exciting figure. You might see it from others in the room. I see Dr. Trainer has just arrived in somewhere, or yes, he is at the back, and I borrowed this from Wim Robrecht, my colleague in Belgium. But really, if we look at that pie explaining the genes linked to familial ALS, we're really making tremendous progress. And if we look at the question mark left there, it's less than 3/4. I'll leave the numbers up to my colleagues who are more expert in that. But frankly, with the discoveries more recently of the TDP43 and fALS, after the initial finding of SOD1, and now the C9orf72, this has been an incredible field, and I think scientists worldwide are feeling like this is a real turning point for understanding the disease. And it's really the familial cases and you as families that lead us to those clues. Again, here it shows you the very well-known now SOD in crystal structure. And yes, frustrating that we identified this in 1993, and our first clinical trials are only coming on board now. But that was the beginning of a field of trying to understand how to work with this information. And we've learned so much since then that I think any new information, and it's testified by the fact that the discovery of C9 has sparked so much research and so many groups worldwide that it's just an exponential effect of having that knowledge now. What it has done for the field, in fact, has developed all kinds of model systems, from looking in a dish to looking at mouse and rats and worms and yeast and flies, and all have their value to us. We can certainly argue whether or not a mouse model which mimics aspects of the disease is going to tell us whether a trial is going to work, but it certainly has given us clues about what happens through the progress of the disease and what kinds of things can we think about in terms of drug targeting. So really our successes and our commitments to the research field has come a lot from genetics, and I couldn't say it better than what you'll hear today from the experts and Dr. Trainer's work, but really these are the clues. And so we spend a lot of our time sometimes with the question, well, familial ALS is only 5 to 10%. What about all of us with sporadic disease? And I think we'll hear more today how actually the impact of the small percentage impacts the whole of ALS. And so this is just an eloquent image of, well, why zebrafish, for example. And zebrafish can tell us a lot because we can see them in live time. They are sort of see-through, and you can image where those cells are that are involved in the disease and look at interesting things on the very right-hand corner where you see those red markings. Dr. Robrecht has actually identified some interesting compounds that seem to change how axons branch, and who knows, that might link into the human disease. So incredible clues come from using these very interesting and dynamic models. And then just to quickly highlight what we do at the ALS Association in our research programs. And actually over the years, I have, and many in the industry, show this pipeline. And in some ways, we sort of limit ourselves by making it the straight line, because actually it's kind of a circle and a process going around, because what comes in and what goes out needs to be improved and come back in again. But in principle, why I highlight this is that having an understanding of the genetics of ALS, the familial cases of disease, identifies for us what we call a target. It's a very complicated disease, and for those of you listening tomorrow, there'll be many more presentations talking about the biology and the complexities. But in essence, the difficult thing for ALS in terms of doing drug discovery and treatments is knowing how to tackle it. And I think their position has significantly changed. One key example is the excitement around the discovery of the SOD mutations. Actually, it was my entree into the field of ALS, where I worked with Don Cleveland to build some of the models. And what we learned quickly from those models is that the challenge is SOD, we understand what it does. It mops up free radicals, it gets rid of toxic waste. But what the mutations are doing is something new. And we still debate exactly about what that new thing is. But what we are able to try and do, and have attempted to do in clinical trials, is to shut down the production of a bad thing. That took some time, but it took us through the steps of having a target, using systems like the mouse model to validate that target, put it into preclinical studies, and take it to clinical trials. The exciting part, and Dr. Benatar will talk a little bit about that, and Dr. Traynor, is the idea that we might be able to use this same technology, a first for neurological diseases. We were the first, now followed by Huntington's and other diseases, to use this technology. We might be able to do this with C9orf, and hopefully with not such a long time frame. So what are we doing at the association with these discoveries? We fund investigators like Dr. Benatar to do clinical trials, and he'll talk to some of his work. We are now funding many groups to build the mouse models around the C9orf, the new mutations. We are also supporting Dr. Benatar to do some of the initial beginnings of trying to work out an antisense study for this new mutation. And unfortunately, tomorrow I had hoped that Dr. Bennett from ISIS would tell us more about the updates of the trials, but he had an emergency and had to cancel. If there are questions about those trials, we would welcome them in the Q&A session. But there's a tremendous amount going on and such excitement in the field, and I'm convinced with your participation and your continued involvement in these trials and providing samples for biomarker studies, we will be able to tackle this, and certainly the industry is becoming very interested in this disease because they can see ways to really develop treatments. So with this, I'm going to introduce the panel today. The speakers will come and speak individually, and if there's time within their session, maybe take a few questions, then afterwards we'll all be up on the panel to address questions. And it is a real pleasure to invite my colleague and investigator and clinician, Dr. Michael Benatar, who has really spearheaded a lot of the important work that is done, including familial patients today. So thank you, Michael.
Sorry, I'm a Mac user. I've got to figure out how PCs work. There we go. Great. Thank you. Thank you, Steve, and thank you, Lucy, for the opportunity to come and work with you today and with ALSA to really expand the time that we have and the array of things that we have a chance to talk about that are relevant to familial ALS. And so I also wanted to thank my colleague Brian Traynor for coming today, and Christine Stanislaw, our genetics counselor, and Natasha Garcia, one of the research coordinators who works with me. And what we're going to try to do is to sort of give you an overview of where things stand. A lot of what we'll talk about, because this is fALS, is genetics, and there'll be some overlap between our different presentations. But I think there's value in repeating or in hearing the same thing over and over in different ways. A lot of it's sort of complex biology. And then I hope we'll have some time at the end. We will have time at the end for some questions and answers. And I know that many of you submitted questions in advance to Steve, to ALSA, and I've tried to take as many of those questions as possible and to touch on as many of those as we can during the course of the morning. So if we've missed any, please do ask them when we come to the Q&A session, and we'll be happy to try to address them. So here are some of the questions we want to try to sort of touch on. What is familial ALS? What's the cause? And Lucy's touched on that a little bit. How common is this? What are the genes involved? What can we learn from twin studies? What have we learned? What are some of the resources that families might be looking for, might be able to use? What's the role of genetic testing, and how do we do that responsibly, including the importance of genetic counseling? What's the relationship between familial ALS and sporadic ALS, and how can familial ALS teach us something about sporadic ALS and vice versa? And then what are some of the research opportunities that are currently ongoing and that might be coming down the pike and of interest to this community? So we'll try to sort of touch on some of this. So I wanted to start with this question of what is familial ALS. This is actually a more difficult question to answer than you might imagine. So first off, we have this artificial distinction, and I say artificial because I think it's increasingly clear that it is artificial, between what's called familial ALS and sporadic ALS. And there's some things we need to clarify here. I think often when we use the term familial, we seem to sort of imply genetic. And when we use the term sporadic, we seem to imply not genetic. But the term genetic really isn't synonymous with familial. Familial just means that the disease is running in the family, that there's more than one individual in the family who has disease. Now, that could, of course, be a genetic cause, that there's something that's been inherited. But you could also imagine if there were an environmental exposure that was relevant and two people in the family living in the same area were both exposed to that environmental factor, that could also produce familial ALS. So I think we've traditionally used the term familial to imply genetic. It's not quite the right usage of the term. And I think we increasingly need to sort of begin to tease this apart. So when you hear familial, yeah, think genetic, but think that it goes beyond that as well. Another sort of thing I wanted to touch on, and I'm sure many of you have seen these sorts of pedigrees. These are what we call pedigrees or family histories. The circles represent women, the squares represent men. When the circle or the square is colored in, that means an individual who's affected by the disease. And when there's a line through the circle or the square, that means the individual is deceased. And so a common question we get is we might get a call, for example, from this person here who'll say, I'm concerned I've got familial ALS. Why are you concerned about that? Well, my mom had ALS. It runs in my family. Well, yeah, that's true, but we don't know that it really runs in the family, right? There's just one individual in the family who's had disease. And so if we're to draw out this pedigree, this family tree, and this is one of the first things we do, at first sort of blush, at first glance, that's not familial ALS because there's only one individual in the family who has the disease. By contrast, if we look at this family tree, if this individual had to call us and say, my mom had ALS, her brother also had ALS, as did my grandmother, we'd say, well, clearly that disease is running in the family, that's familial. We think there's a high likelihood of that being genetic, and that's the first thing we go after, but there may also be environmental factors that contribute to that risk. And so there's sort of this gradation or the spectrum between having just one individual in the family to two individuals to many, many individuals. And the genetic component may sort of increase the more people there are in the family who have, the more people who have the disease, perhaps the greater the likelihood that there's a genetic contribution to that. And I will say, sometimes we see what I've not drawn out here, is somebody might call in and say, you know, my mother had ALS, and if I look at my husband's side, his great-grandfather had ALS, and is that familial? Well, that's actually ALS on two different sides of the family. And I guess, yeah, in a sense, that's familial, but that's not genetic, or at least we don't think that's genetic, because those are two different genetic sort of populations or groups that have come together through a marriage. So that's something that we sometimes see. So, are sporadic ALS and familial ALS different diseases? I would say that clinically they're indistinguishable. If we as clinicians see somebody who's got familial ALS or sporadic ALS, they look just the same. The symptoms are the same, the physical examination signs are just the same. And I will say this, that all the genetic mutations that have been found in families with ALS have also been found in people with sporadic ALS. Maybe not with the same frequency, but they've also been found. If we look at first-degree relatives of people who've got sporadic ALS, they're at a slightly increased risk of developing ALS than first-degree relatives of people without ALS. So maybe there's a genetic contribution there as well. So I think that we have this classification. I think it's convenient, but I think it is to some extent artificial. I think the biological meaning is limited, and I think some of the new genetic discoveries that Dr. Traynor is going to talk about sort of help to emphasize sort of the artificiality of this distinction. It is useful, it helps us sort of get traction and make sort of progress scientifically, but I do think to some extent it's an artificial distinction. So how common is familial ALS? So if the overall incidence of ALS is 2 per 100,000, now what do I mean incidence? I mean the number of new diagnoses per year. So about 2 people per 100,000 in the population develop ALS. So the number of new diagnoses of ALS per year in the US is around 6,000. So if fALS accounts for about 5% of that, that means that there are about 300 new fALS diagnoses every year. So it's a good number, but it's not a huge number when we're trying to study this problem and involve people with these diagnoses in research studies. So what about the prevalence? Prevalence is the number of people, if you just look out into the community or to the population at any point in time, how many people are living with the disease. It's not the number of new diagnoses, but the number of people who just carry that diagnosis at a point in time. And that's about 6 per 100,000, which means the number of ALS patients in the US at any point in time is about 18 to 20,000 ballpark. If fALS accounts for 5% of all ALS, then the number of fALS patients in the US is around 900. Now, one of the things we know is that if you look at a familial ALS pedigree or family, there isn't always somebody living with the disease at a given point in time. There may be a history of people who've had the disease but who have passed away from the disease. And so if about 75% of families have somebody living with the disease, that means we think there are probably around 1,000 or 1,200 pedigrees or families living with this disease in their families in the United States. So that's a good number, and that's an estimate. We don't know that that's absolutely correct, but that's sort of our best guesstimate of what these numbers are. But I also think that we may be underestimating the frequency of familial ALS. Often when people are seen in the clinic, the family history may be incompletely recorded. You may be talking to an individual and they've lost contact with part of their family. They don't have complete information about the family. So you may be sort of getting incomplete information. There may be misdiagnosis. A family member might have been told they have multiple sclerosis or lead poisoning or spinal muscular atrophy or something else. And so we may be underestimating the frequency of a family history of this. Sometimes, I think this is less true in ALS, maybe more true in other diseases like Huntington's, there's perhaps a reluctance to report the family history. Sometimes if the family size is small, or if there have been early deaths due to other causes, for example, childhood deaths, that might have been somebody who might have been at risk to develop disease in the future but never did. And so we may be underestimating the frequency of familial ALS. And then there's this important issue of disease penetrance that may be incomplete. And I'm going to come to that and what that means. But that's a very, very important thing. And I'm quite sure that Dr. Traynor and Christine will comment on this issue as well. So what's the cause of familial ALS? And I've said, I think largely we think this is genetic, but we shouldn't exclude there being an environmental component to this. We just don't know what those environmental factors might be, and so I'm setting those aside for now. But DNA is the genetic code. This is the material that we inherit from our parents. You've heard of this double helix. This is the picture of what this typically looks like. And this is sort of made up of a code. Think of it like, the alphabet or your telephone number. It's a code. And a gene is just a stretch of this DNA. So long stretches of this DNA make up chromosomes. And if you take a short stretch of that DNA on a chromosome, that's a gene. And when that code is sort of mistranscribed or misspelt, it's like you're jotting down somebody's phone number and you miss a digit. Right when you now dial that number, you don't get through to the person you were looking for because just one of those digits is off. And so if we get a genetic mutation, a change in the gene, That's what we think then leads to familial ALS and many other genetic conditions. So for the purpose of this discussion, fALS is caused by DNA mutations in specific genes. So this is a slide that Dr. Brown from the University of Massachusetts kindly shared with me, and I wanted to sort of share it with you, but I think it shows the incredible progress that's been made. If we look back in 1993 when the SOD1 gene was described, this is the number of genes that we know, and this is time. Here it was one gene. Where are we at now with C9orf72 in 2011? We're at about 16 genes. So you can see this is exponential progress that's been made in terms of uncovering the genetic basis for familial ALS and for ALS more generally. So I think Lucy and I must have been making the same slides or thinking about things in the same way. I'm pleased to say I think our numbers are much the same. And so, these are approximates. I don't think we have perfect numbers. And Dr. Trainer may show slightly different numbers, but the impression is the same, is that we now know a large part of the pie. We know what the genetic cause is, probably in around 6 out of 10 people. So perhaps in still 4 out of 10 people, we don't know the genetic cause. But that's huge progress. If we'd stood up here just a couple of years ago, we would have said we know the cause in perhaps 2 out of 10 people. So we're making great progress, and that's thanks in large part to your engagement, your involvement, your active participation in the scientific process. And of course to the hard work of the scientists. But this is a collaborative process that can't be done alone. So I did want to say a word about how familial ALS is inherited, because I think this is important and will be an important foundation for some of what Christine will talk about when she's counseling people about having genetic testing and what do the results mean. And so broadly speaking, we make a distinction between what we call dominant inheritance and recessive inheritance. Let's talk a little bit about what each of those mean. Let's start with the dominant inheritance. I'm going to start with that because we think that most of the genetics in familial ALS is dominantly inherited. Most of it. Not all of it, but most of it. And certainly most of it in the United States. In Europe, there's a little bit more that's recessively inherited. So what dominant inheritance means is that we've all got 2 copies of a gene, right? We inherit a copy from our mother and we inherit a copy from our father. So we've got 2 copies. What dominant means is if you have a genetic change or a genetic mutation in just one of those copies, you can get the disease. By contrast, recessive means you need to have 2 abnormal copies. You need to inherit an abnormal genetic copy from your mother and from your father in order for the disease to become apparent. And what that means is that when it's recessive, even if you've got 1 genetic abnormality, if you've got 1 normal copy, that normal copy is fine. You can get by with that. It doesn't matter to you that you've got that abnormal copy. So here if we look at the dominant, the way this sort of figure is sort of drawn out is this individual, the male, it just happens to be the male here, he's affected. He's got 1 copy of the affected gene and 1 copy of the normal gene, but because it's dominant, he's affected. And here's his wife. She's got 2 copies of the normal gene and she's unaffected. Now let's imagine they have 4 children. And what they can do is they can pass this gene on at a 50% risk. So if we just sort of parcel this out, the affected gene can come to a son, the affected gene can come to a daughter, the normal gene can come to a son, can come to a daughter, and the mother can do the same thing. And if you sort of look at that, you'll see that 50% of the children, there's a 50% risk of inheriting that gene. And if having the gene, just 1 copy means you can get the disease, there's a 50% chance of getting the disease. Now that sets this issue of penetrance aside, and I'll come to that in a moment. But when the disease is recessive, if you have just 1 copy of the abnormal gene, that makes you a carrier. You carry the gene, but you don't have the disease. You can still pass that gene on to the kids, but now there's only a 25% chance that a child will inherit the abnormal gene from the mother and the abnormal gene from the father, and then have 2 abnormal copies of the gene and go on to develop disease. So that's important. When we're talking and doing genetic counseling for familial ALS, by and large, this is the paradigm that we're talking about. So here's this issue of penetrance. Now, penetrance is sort of a technical term. What it means is the number of people who have the gene who go on to develop the disease. So if 100% of people have the gene and 100% go on to develop disease, we call that complete penetrance. The gene is completely penetrant. It manifests, it shows itself. On the other hand, if 100% of people have the gene, and let's say, for example, only 80% of people go on to develop disease, we say that penetrance is incomplete. Now, that could be 80, that could be 60, that could be 30, that could be 10. And as we make genetic discoveries and learn about more genes, we have to figure out what that penetrance is. But that's very relevant. You might have a 50-50 chance of inheriting the gene if it's dominant inheritance. But if the penetrance is only 20%, doesn't necessarily mean that having the gene means you'll get the disease. So that's stuff we have to figure out and be able to communicate to people when they go through genetic testing and genetic counseling. So is genetic testing possible? Yes, it is, but there are caveats. So the first thing to say, and people often ask us this, is I know that my mother had the SOD1 gene, but what gene will I have if I have the gene? And by and large, and I want to sort of say this, and there are always exceptions, but genes run in families. That's their nature. You inherit them. So if the SOD1 gene is the gene that causes ALS in your family, then if you're going to be at risk, or if you are going to have the gene, you're going to have the SOD1 gene. Now, of course, you can always come up with a counterexample. Two families with different genes might intermarry, and there might be more than one gene in the family. But by and large, it's one gene that runs in the family. So we need to recognize that. Now, genetic testing should always start with an affected individual. We need to start with somebody who we know has the disease. And the reason for that is we have incomplete genetic information. Remember, we said to you, Lucy said, and I said the same thing, that in 40% of people, we don't know the gene. So if we don't know the gene in the family and we start testing unaffected people and you don't have any of the genes we know, well, that doesn't mean you may not be at risk for developing disease. Maybe you have one of the genes we haven't yet found. So we first have to know what the gene is in your family, in an affected individual, and then we can tell you whether you can have pre-symptomatic genetic testing and what the results of that testing might mean. And this is where genetic counseling becomes very important. So what about pre-symptomatic genetic testing? Well, again, only if the gene is known in the family. It's important to be clear about the motivation for undergoing genetic testing. Why do you want to do this? It's important that people are ready to receive this information. This can be stressful information. And really understanding what does it mean to have this information? Why is it important? Why do you want to do it? Can you live with that information? What are the implications for other family members? And Christine, I think, is going to go through a lot of this during her talk. And so genetic counseling is really, really important here. And this is something we offer as part of our pre-fALS, our pre-symptomatic familial ALS study. But this is also commercially available. You just need to make sure that you're getting it if you're going through testing and you're getting it from somebody who knows ALS, who knows ALS genes. So there was a specific question somebody asked about twin studies, and I'm not the guru, and maybe I'll defer this to Brian to talk about or to answer in the panel. But twin studies can tell us something about what's called heritability. It's a measure of the relative contributions of genetic and non-genetic factors to how much there's what's called the phenotypic variation, how much difference there is in the manifestation of disease within families. And heritability in sporadic ALS, so how much there's a genetic contribution, has been estimated to be around 60%. So what's that saying is that amongst families where the gene expresses itself and you develop disease or you don't develop disease, about 60% of that variability may lie in the genes. It may not lie in a single gene, it may lie in a constellation of genes, but it suggests there's an important genetic sort of underlying risk for developing disease. And so that's important. Now, people had asked about whether there's information about familial ALS in registries. Now, you all know that there's a national ALS registry, and I encourage you to sign up for this registry. And this does document the presence or absence of a family history of ALS, as well as a family history of other neurodegenerative diseases. But it doesn't go further than that, at least not yet. And we had set up something related. It's not to be instead of, it's in addition to, a familial ALS registry. This is for people who have the disease, but also for people who are at risk for disease. And I encourage you to go and check this out at falsconnect.org. And I think many of you in the room have signed up for this already, and this is something we're trying to grow and to use as a tool to communicate to you new information and new research studies as they become available for you to participate in. And if you go to the business snapshot from the web page, and Deb, you'll recognize one of your photographs that you sent me. And if any other people have photographs, please do send them. We'd love to put them up here. But you can go here. There's some information about familial ALS, about research studies that are ongoing. And you can click on a button up here about research opportunities. And that'll then tell you about the pre-fALS study, about ongoing clinical trials, about efforts to look for new genes. We're always encouraging people, if you know of studies we don't have listed here, if you're participating in one, let us know. We'd love to stick it up. We're always asking other scientists to send us information. We would like this to be as complete as possible. And I'll say a quick word about each of a couple of these studies. So pre-fALS, as some of you may know, is the pre-symptomatic familial ALS study. This is enrolling people who are at genetic risk for developing ALS. So we start with families, we identify the gene, and then we look for healthy people in those families who may carry the gene, so that we can try to study this disease before it becomes apparent. We think the disease process may begin sometime before the disease becomes apparent. And maybe once it becomes apparent, it's almost too late, and we have to get in early. And so we're trying to study this disease as early as we can. So we want to characterize pre-symptomatic disease. We want to find out what's the annual risk of people who are at risk going on to develop disease. And that's important if we want to do a preventative trial. If we want to start a therapy early to prevent people developing disease, we have to know how quickly people go on to develop disease. So we're trying to figure that out. We're trying to figure out why some people with the gene get the disease when they're 25 and some people don't have it when they're 75. So trying to figure out what some of the genetic and environmental factors are that modify this risk is an important goal. We don't have time today. Maybe in the Q&A session we'll talk a little bit about biomarkers. These are things that we can measure that help tell us something about the disease. And for those of you who have participated, you know that we collect blood, we collect spinal fluid, we collect other biological samples, that we can do this kind of discovery to look for markers of the disease that might be helpful in developing therapies. And then we want to use the knowledge that we gain to try to design early therapeutic or even preventative trials. I'm going to skip that because I think I'm running over time a little. Some of you may know about this trial of arimoclomol. This is what's called a phase 2-3 trial. So this is a trial that's looking both at safety in phase 2, but also trying to look at the efficacy of this drug. How effective is it in phase 3? And this is enrolling people with rapidly progressive ALS due to specific mutations in the SOD1 gene. This is a trial that's jointly funded by the ALS Association and the Food and Drug Administration. It is a randomized, placebo-controlled trial. The dose that people are receiving is 200 milligrams 3 times a day, and the treatment duration is 12 months. Now, we're just nearing completion of phase 2. We have, I think, about 28 people enrolled. We need to get to 30 or 31, so we're getting close. And so we expect that we'll be able to do this interim analysis within the next 9 to 12 months. Why so long? Because it's not just getting 31 people in. We have to have a period of follow-up of those people. So we hope within the next year that we'll be completing that analysis and then hopefully moving on to phase 3. There's also a trial that Dr. Dale Lang in New York is running of a drug called pyrimethamine. That's an anti-malarial drug. And this is in slightly earlier development. It's in a phase 1-2 trial. I had emailed Dr. Lang to try to get an update on this to give you as current information as possible, but didn't hear back from him in time for today. So I apologize if my information is a little out of date. But he's aiming to evaluate the safety of this drug and the extent to which it suppresses spinal fluid levels of SOD1. The idea being that when there's a mutation, if those levels are high and if he can suppress those levels with this drug, maybe that's a sign that the drug would be effective in treating the symptoms of the disease. So he's looking for people with familial ALS with any mutation in the SOD1 gene. He's using up to 75 milligrams a day, and the treatment duration is about 36 weeks. As far as I understand from him, there's no placebo here. And you can look this up on clinicaltrials.gov, and there's some contact information there to reach out to him if you want some more information. I did want to say a word about this. I know that Dr. Traynor is going to talk a lot more about the genetics of this, but just to say that this now looks like the most common, at least identified to date, cause of both familial ALS and sporadic ALS. And based on the nature of this genetic mutation, it's very amenable to therapy using this thing called antisense oligonucleotides. And as you may know, ISIS Pharmaceuticals has already brought this into a phase 1 trial for the SOD1 gene, and they have efforts now to do this for the C9orf gene. We also have a small pilot study funded by ALSA to develop this approach at the University of Miami using a slightly different biochemical approach. And so these are efforts that are ongoing. These are still preclinical. They're not yet trials, but your engagement and your participation is still crucial to the development of these trials. And so if you have questions about that, please do get in touch. So I know there are also many efforts ongoing to identify new genes. We still don't know the genetic cause in 40% of people with familial disease and the genetic contribution in many people with sporadic disease. We need to figure that out. There are many efforts ongoing. Dr. Brown at UMass, Dr. Sadiq at Northwestern, Dr. Traynor here at the NIH, and many other groups, both in the US and in Europe. And so, you know, if the gene in your family is not known, there's a great opportunity there to help contribute to science, to discovering that gene and how that might shed light on the biology, but also potentially lead to development of trials and treatments that are specifically focused on the gene in your family. So when you're choosing a study, and this is important, I want you to think about this, talk to the investigators. Understand the rationale for the study. What is the study trying to achieve? What's it trying to learn? Always be informed. Understand what are the study procedures, what are the potential risks, what are the potential benefits, and perhaps most importantly, ask questions. Your questions are tremendously valuable. They should and do help us as scientists decide what's the next step. How do we design the study? How do we modify the study? How do we make sure that we're designing studies and running research in a way that engages you, that is relevant to you? So I think we need more of your dialogue, more questions from you, and I know ALSA feels the same way. So however you feed those questions directly to the scientists through ALSA, we need to hear that. We need to hear that from you. I saw this cartoon in the New Yorker recently. There's a cure, but it's light years away. That's not how we feel. We don't want this to be light years away, and all of this work that we're doing is trying to make sure that a treatment is coming soon. And I don't want to overpromise, but science is making progress, and I hope that as some of these pieces of the puzzle fall into place, that suddenly a picture will emerge and real meaningful therapeutic advances will take place. So I wanted to thank the ALS Association, one, for all their efforts, but particularly for organizing Advocacy Day and particularly for organizing this fALS Summit and for all their efforts. I want to acknowledge the myriad of investigators and their research staff at institutions all around the country who are engaged in this research. This really is a community-wide endeavor. This is not one individual or one research program. This is many people coming together. I wanted to thank you for your participation in Advocacy Day and all of you who participated at any level in research, because none of the progress could be made without you. And I want to leave you with a little bit of contact information, and if I have time, I'm happy to take a question or two now, otherwise we'll hold them for the panel. I wasn't timing myself, Lucy. I'm not sure how. Okay. Okay, so we'll hold them for the panel. Thank you very much.
Thank you, Dr. Bennett. That was really very clearly explained and will set the stage nicely for the complicated story we're about to hear from Dr. Traynor, who is going to give us the new updates. And he needs little introduction. I know he's spoken on webinars, and we feel very excited that he's made such an incredible contribution to the field of ALS and really at a turning point now in understanding what's going on. So thank you, Dr. Traynor.
All right. All right, well, good morning, everybody. I'm going to multitask here for a second. And let's see now. It should probably work. Okay. Hopefully it's opening up. All right, well, it's an honor to be invited by Lucy and by Michael to come and talk to you this morning a little bit about how we're progressing in terms of our research in ALS, and particularly with respect to the genetics of this condition. I think that the last year has seen some tremendous progress, and I don't think that either Michael or Lucy have been overstating it. I think that really there has been a tremendous sense of excitement within the community in the last 6 months, because I think that now for the first time we're beginning to piece together not just the cause, but potentially, and I do want to emphasize the word potentially, over the next number of years, maybe even a mechanism for going after this and stopping it in its tracks, one hopes. And this is certainly something that I'll come back and talk about during the context of my talk. Now, I want to start off with something that, I do this for a living. I look for the genetic causes of ALS, but every so often I get thrown a curveball, either by a scientist or by a patient, and they say, well, why are you bothering with this? Why do you spend so much time trying to find the genes that are responsible for this disease? And sometimes, kind of go, wow, that's such an obvious question. I haven't really thought about it. But there are, when you sit down and try and go through the reasons, there are some very obvious ones. I think that from the perspective of the patient, knowing the genes that are responsible are very important in terms of helping us with diagnosis. I think that not everybody here in this room will appreciate this, but some of you might, that at the start of the disease, sometimes even the best neurologists among us struggle to make the actual diagnosis. And having a test, a genetic test, would help us in that regard. It sort of removes the uncertainty to a certain extent and helps the neurologist and the patient and their families move forward in their understanding of what's going on in their bodies and why they're getting weak and so forth. From a scientific perspective, it's very important for us to understand the disease. Why is it that motor neurons die? Why is it just the motor neurons die? Why is it so specific? Why is it so selective? And why is it so rapid in terms of its progression? And understanding what it is, what cellular processes underlie that, and what pathways, what common pathways underlie all of these things is really important for our understanding of the disease. Why is it important to understand the disease? Well, by understanding it, it improves our chances that we'll be actually able to do something about it. One of the key things, and of course Lucy has been instrumental in this, this is what Lucy did before she joined the ALS Association, is animal models of disease. Basically taking those genes that we identify and putting them into different types of animals, usually mice, but you can also do it with rats, you can do it with flies, and you can do it with worms indeed. And the reason why that's important is not only does it tell us a little bit more about why the cells are degenerating or why the cells are dying, but it also means that we can actually treat those animals with agents that we think might work. And see whether they have a beneficial effect. So rather than introducing agents for the first time into humans, you try them out in these animals first and foremost. Because you want to get a sense of whether they're toxic, you want to get a sense as to whether they work or not. And then finally, and I think I left this deliberately to the end because I think it's probably the most important aspect of it, is knowing the genes gives you a target, gives you something to go after. If you know that a motor neuron is degenerating, great, you can have a shot at trying to find a small molecule or something that will go after that. But if you know exactly why that neuron is dying, and you know exactly precisely the molecule that's involved and the protein that's involved in that dying, then you actually have a better chance of being able to find something that reverses it or slows it down. And I think that really is still remains the challenge to us, that as you'll see as I go through these slides today, the technology for genomics has improved tremendously. Really, it's light years ahead of anything that we even dreamt of 5 years ago. But we still struggle a little bit in terms of developing the therapies. If there were drug companies here in the room today, they would tell you that usually the time it takes from the identification of a protein that's involved in disease to the actual first clinical trial, when the drug first hits human beings, is about 10 years. And the vast majority of of drugs that are in the pipeline never make it to humans. They actually die before that. They are actually killed off before that. So I think that that particular, we can't overstate how difficult it is, that almost we've got to the point now that finding the genes is easy, but finding the cure for it is a little bit harder. And I think that's an important point to make. So you've seen that slide from Bob Brown that Michael showed, basically about how basically the history of discovery of genes in ALS. Now, one of the reasons why I'm putting this up here, not to go through each and every one of them, but rather to show you that the first gene was first described, that was SOD1 back in 1993. Then there was a very long hiatus until about 2008, 2009, and then a whole bunch of other genes started to come. And then even within the last 6 months, last 12 months, we've had 4 new genes being described. And I think that that shows to you that the pace of discovery really is improving. Now, why is that? Well, it turns out that because of the efforts of the Human Genome Project, funded by NIH and by others around the world, the sequencing technology, our ability to sequence genomes, has really improved to the point that now what we talk about, the $1,000 genome, is almost a reality. And in fact, we're not even talking about the $1,000 genome anymore. We're talking about the $100 genome. And this has really driven our ability to find things. I won't say that it's made it completely easy. It's never completely easy. Each new technology, no matter how wonderful it is, carries its own set of caveats and problems with it that you have to understand and you have to be familiar with. But certainly nowadays the landscape is much easier to navigate than it was before. So what I want to talk for the rest of my talk about is our most recent gene that has been found in ALS, and it's a gene. It's actually the gene is called C9orf72, and what that actually tells you is that the gene doesn't actually really have a name as of yet. It's called C9 because it's on chromosome 9, ORF because it stands for open reading frame, and then 72 because it was the 72nd one on chromosome 9 that was found. And the reason for that is because this is such a new protein, such a new gene, that we really know very little bit about how it works. We just know that it underlies a tremendous amount of familial ALS and sporadic ALS. So we call it C9orf72. Now, this particular gene sort of took on the aura of holy grail within the ALS research community for 2 reasons. First and foremost, it was obvious from a very early stage back in 2006 when it was first sort of its existence was glimpsed. It became obvious that it accounted for a large percentage of familial ALS cases. And the other reason was, despite all of the advances in technology, nobody could find it. And this is despite the fact that there are many neurogenetics laboratories all around the world, very, very good places, working very, very hard on trying to crack it. And we realized this from an early stage. So what we said was, okay, let's reach out to all of these different laboratories around the world, some of which were actually our competitors. And we said, okay, well, we've got the resources, the sequencing resources here at NIH. What you guys have, you families that you've collected, individuals such as yourselves who've agreed to participate in research that are linked into chromosome 9. So you send us your samples, we'll sequence them, and we'll send you back the data, and we'll analyze it together. One of the main families that we found was this family right here at the bottom called the Gwent1. Maybe a little bit small to read it, but it's called the Gwent1 kindred. It's called the Gwent1 after a small little village in the wee valleys of Wales, in just outside of Cardiff. Actually, it's located, if I had a pointer, I'd be able to, maybe I can use the mouse. This is Cardiff here, and actually it's just a little bit north of Cardiff. In fact, it's such a small place that I'm told, I haven't been there myself, but I'm told if you blink as you drive through it, you'll miss it. But anyway, a very talented neurologist by the name of Hugh Morris had spent a decade collecting samples from this family in this small little village that had basically ALS and frontotemporal dementia running through them. And we recognized that this was a very important family from the beginning. Now, we analyzed it and we sequenced it in a variety of different ways. But in the end of the day, what really worked for us was we took a cell line made from one of those individuals and we sent it off to a company in Germany. And they sorted out just chromosome 9 for us. So they sent us, but we sent them cell lines and they sent us back DNA that was enriched just for chromosome 9. Why was that important? Well, what it allowed us to do was to sequence the entire chromosome 9 to a very, very deep level, basically up to 400x. That's a technical term. Suffice to say that generally we look for about 10x coverage in our genomes. And we were up to 400x, so we were about 40 times sequenced every single base pair. I'm not going to show you the actual thing that we found. It's a little bit hard to understand, but suffice to say what we found was that there was a 6 base pair, 6 of those letters that Michael talked about earlier. We all, normal individuals, have about between 3 and 6 of those copies. So it's basically 6 base pairs, G, 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 C, C. And it's in a normal individual has them about 6 times, up to 6 times. And what we found was that ALS patients had them hundreds, if not thousands of times, repeated again and again and again and again. Now you can imagine that the human genome is a very sensitive sort of area. It's very precise, it runs like a metronome, and having a big chunk of G's and C's stuck in there, which aren't supposed to be there, upsets things and causes the cell to become unstable and to degenerate. Once we found it, we went back to make sure that we were right. We went back to various families that we had collected in collaboration with others in order to see whether it's segregated with disease. And again, these are pedigrees here. They're a little bit small to see, but I think that the point to make here is not to show the individuals within it, but rather that there's a large number of them. There are several there. The only reason we were able to collect that many families was because A, good graces of yourselves around the world, individuals who believed that research and finding out what the cause of this disease is important. And secondly, because we were able to collaborate with a large number of institutions and labs around the world. One of the things that I should mention is that I'm just back from Finland. I actually got back at about 12 o'clock last night from Finland, so I'm probably still asleep somewhere in my head. But the reason why I was in Finland is because it turns out that this particular expansion, this particular genetic mutation, is extremely common in Finland. And actually, we had used the Finnish population in order to find it. And we knew that when we went back, if we found the mutation and went back and looked for it, that it was going to be present to a large extent within that population. And lo and behold, when we did, we found that 50% of all familial ALS cases in Finland carried the expansion, carried this mutant gene, which is, a sizable proportion, you can imagine. And the reason why Finland is so good for studies like this is because it's a highly conserved population. It's up there in the north of Europe, and there's not so much population migration. And having just come from there, and it's May, and it's freezing and it's cold, I can understand why there's not that much migration up to Finland. But now we knew from the beginning that basically Finland was going to be a high rate within the Finnish population. What we weren't expecting was what the rate was going to be in the more outbred European population. What do I mean by that? I'm talking about Italy and Germany and the United States. And to be honest, I thought that if we saw it in 5 to 10% of those cases, that we would be, that was a big deal, because in historical terms, that was a huge percentage in terms of ALS. And I remember as the first plate was coming off the sequencer, coming off the machine for Italians, it was actually Italian samples, familial samples, every second sample literally carried the expansion. And those of you in the audience who know me know that I'm very seldom at a loss for words. In fact, it's hard to shut me up sometimes. But I think I was flabbergasted at that stage. I never thought I'd see a day when one mutation would cause or be responsible for such a large proportion of familial disease. Ended up accounting for about 40% of familial cases within the Italian and Germans. This gave rise to 2 publications back to back, one from our group and our international collaboration, and one from Rosa Rademacher's at the Mayo Clinic, published in Neuron only last September. So we're talking about 6 months ago. And since then, it's really, as I said before, taken off. And I think that's highlighted by the fact that it's already been cited by nearly 50 papers on this. And that really shows how important and how interested people have become in this. So what does that mean for us in total? You've seen both Lucy and Michael have shown you pie charts. I'm going to do exactly the same. The figures are roughly the same. So just, I more summarize things rather than showing the individual genes. I think that right now, we know about 2/3 of the cause of familial ALS. It means there's still 1/3 out there to find. All right, so about 1 in 3, we don't know the actual cause. Interestingly enough, when we look in sporadic cases, we also find the same mutation. And for reasons that I will not bore you with, it is possible that those sporadic cases are actually distant family members of the familial cases. They just don't know about it. And that now accounts, we now understand about 11% of sporadic disease. And when you add them together, that means that in total, we understand about 15% of all ALS. That's a great step forward, but I think that everybody in the audience will realize, well, wait a minute, what about the other 85%? And that is important for sure. And that is what Michael is working very hard to try and understand, what I'm working very hard, and what Lucy is working very hard to raise the money to fund the research and to choose the right research projects. Because believe me, one of and I'm sure Lucy will agree with me when I say this, there's no shortage of research projects out there to fund, but you have to pick the right ones. And that, I think Lucy should be praised because she has done a tremendous job over the last number of years in very savvy picking the right investigators, the right labs, and the right projects to move forward with. This is the bit that reason why I like genetics. Finding the genes is one thing, but sometimes you can have a little bit of fun with it. So if you look at the distribution of the mutation across the world, it looks like it's very high up in Scandinavia and then decreases as you move down and then decreases as you move across the rest of the world. And this has given rise to our theory that it was in fact the Vikings. That it started, there was one mutational event about 1500 years ago, about the time of the fall of the Roman Empire. And then that occurred in Scandinavia, and then the Scandinavians brought it on their the Vikings brought it on their summer holidays as they went around Europe. Of course, it's a difficult theory to prove or disprove, but right now it's a nice one. I also particularly like this slide, Vikings like pirates but cooler. This is a map showing the distribution of the Vikings around Europe. This is where they actually conquered. And actually, it's remarkable looking at this. I was actually, it really struck me when I was looking at this, because let me show you that this area here is about the same, and this is where Paris is. I don't know if you can see my arrow right there, okay? So we were collaborating with a very important group in Paris, and they were ringing us up, and they were telling us, Brian, we're seeing this in a huge percentage of our cases. I was kind of going, that's kind of, even by C9 orph standards, that's unusual. And then we were contacted the same day by our collaborators in the Netherlands saying, Brian, we're seeing it in such a small percentage of our cases. We don't know. We only seen it in about 2%. So it turns out that if you think about the Vikings in historical terms, that actually makes sense. Because the reason why the Vikings were successful militarily, what their tactics were, was they stayed along the coasts. And then they had shallow draft boats that they used to sail up the rivers, attack the cities and towns on the banks of the rivers, and then take the loot and go back down. And it turns out that they started going up the Seine, all the way up the Seine River to Paris. And the French king at the time, don't ask me which one it was, decided that what he'd do is try and buy the Vikings off. So he got 2 large chests and he filled them full of gold. And he gave them to the Vikings and says, now, go away, leave us alone. And the Vikings, in their infinite wisdom, said, these guys have gold. And they started attacking even more. Hence why we think that around the Parisian area, there's such a high percentage. And then in contrast, if you look at the Netherlands, the Dutch king at the time, he fought the Vikings and he kept them out of his country, which might explain why the rate is so low in that part of the world. So it's, you know, from a scientific and from an understanding of the disease, this sort of stuff is kind of cool. So here's, I like to be provocative. I think that this is, I'm putting this up here to be slightly provocative, but I also believe firmly that this is true. I think that ALS genetics is solvable within the next 5 years. I think that in 5 years' time, that pace of discovery is going to continue. And I think at the end of those 5 years, we're going to know the cause of virtually all, if not all, ALS. Now, does that necessarily translate into being able to treat it and to be able to cure it? I hope so, but there is no guarantee about that. Okay? And that's going to be done through a combination of exome sequencing, genomic sequencing, and large-scale GWAS. If anybody wants to know further about that, you can certainly ask me at the end or come up to me afterwards, but it's not something that I'm going to go into details about. I'm going to give you, this is sort of a very flagrant plug. We still need samples. There's still, you know, there's a lot of work to be done. And there's my contact details, Brian Traynor, trainorb@mail.nih.gov. Sorry, it's in such a horrible colored font. We are organizing, thanks to Lucy and to Stephen and to everybody. We have the, as we've done for several years now, collections for samples for tomorrow. We particularly want anybody who has familial ALS, but also we will be very grateful if anybody with sporadic ALS will come to us. And I'm going to end with this slide. Now, I usually when these slides come up, people start talking about all their fellows and their postdocs who did all the work in the lab, and believe me, that is true. But that's not the reason why I've put this particular slide up. I've put it up for two reasons. The first and foremost is, look at the number of names that are on that slide. All right? Gone are the days of science being the permit of one individual lone scientist late at night time in the lab. We now are into the era of international collaborations. And I think that that's what this represents. And of course, Michael Benatar's name is there as well, because we collaborate with Michael. And then the other thing I want to say is that we are funded. We are very fortunate to be funded at NIH by the federal government. It's a branch of the US federal government. And believe me, that we have deep pockets, and we are funded there. But what we do is very expensive. And quite frankly, we would not be able to do these projects without the support of the ALS Association and other charitable organizations, which I know that all of you are very active in. And with that, I'll stop and take any questions that you might have.
Thank you so much. Hold your good questions. Thank you for a tremendous presentation. And we're certainly very pleased to be able to support Brian's work in combination with many other collaborators. We do think that these kind of discoveries, and particularly this one, is going to change the field of ALS. So last but not least, it is my pleasure to welcome Christine Stanislav from Emory University and in Michael's team, a very, very important aspect, and so pleased that she's part of our panel today. Thank you.
Hi, good morning. It's great to be here. Michael actually slipped away to Miami when I was out on maternity leave, so I've been doing most of my work by telephone for the last year and a half, so it's great to see actual faces here in front of me this morning. I'm going to hit on some things that have already been touched on, but again, I think they're important points. I sort of approach this by thinking about over the 4 years that I've been working with the pre-fALS study, what sort of questions have I received from people, what kind of misconceptions have I found, and tried to address some of those issues. So back to a little genetics 101. Genes come in pairs. We have probably 25,000 different genes that control all aspects of how our body grows and develops and functions from day to day. And each of those genes comes as a pair, one that we get from our mom, one that we get from our dad. And we all have all of the genes. So, one of the first things that I found is that not everyone realizes that when we talk about some people with familial ALS have problems with SOD1, some have problems with other genes. All of us have SOD1. All of us have all of these genes that people have been talking about. It's just a matter of kind of what form are they in. Are they functioning normally, or do they have a problem with the DNA structure, what we call a gene mutation, that's going to lead to increased risk for disease? So how do we test genes? If anyone here has gone through genetic testing, you know that it's pretty easy in terms of what kind of samples we need. We don't necessarily need tissue samples or anything crazy like that. It's usually just a blood sample. Sometimes it can be done on a saliva sample. But what are we actually doing in the laboratory with that? We're actually, in a lot of cases, and the scientists here can correct me if I'm wrong, but most genetic testing these days, when we're starting out with the very first person and we're seeing, is there a change anywhere in the gene? Because all of these genes, for the most part, have multiple different mutations that have been found. So again, not all families with SOD1 mutations have exactly the same mutation. So when we're starting out, we have to figure out what's going on in a particular family. Do they have a mutation in this gene? And if so, what is it? So typically that's done by a method, what we call gene sequencing, when we're actually going here. Let's see, do I have a, there, yep. We're going here, sort of isolating out from the individual chromosome location where the piece of DNA that corresponds to that individual gene is. They can take it apart in the lab, look at how all these little pieces, all these A's and G's and T's and C's line up, make sure everybody's in the correct order, make sure everyone's in the right spot. And there are lots of different ways that gene mutations can happen. Sometimes it's one letter change for another, sometimes it's these big expanded repeat segments that you just heard about that are causing problems. But that's really where we have to start. So which gene do we test? And that's the tricky thing, and that gets us back to, sort of who do we test and why? Now this is sort of Norman Rockwell's depiction of a family tree here. That's not normally what we see. I usually get things more like this, which you've already seen pictures of today. But as was pointed out earlier, it's very important that we actually start by doing testing on an affected individual, if that's at all possible, because if we get that unaffected person coming in very concerned, and very appropriately so, about their family tree, we've got this whole long litany now of potential ALS genes. We don't know where to start, and we don't know if we test them all and find nothing, if that really changes that person's individual risk. There may still be risk there, or maybe there was a gene mutation, maybe there was a common mutation in their affected family members, but we unfortunately don't know that, so we can't adjust their risk. And that's one really important thing to think about in terms of genetic testing. We also now have the challenges that we face with all these new genes that have been discovered that we're only just now learning about. We're learning about how they function within families, what kinds of things might we expect. It may be that within a given gene, some mutations have a little bit of a different course than other mutations within that gene. And again, those are things that we're going to learn about over time. Again, do all mutations carry the same risk? Could there be interactions between genes that are going to influence disease risk, age of onset, symptoms, all of those sorts of things? What we know about SOD1, is that really going to be applicable to families that have mutations in other genes? And so we're still a little bit limited in how we can interpret some of this information that we get out. So testing unaffected relatives, and this is a big question. Certainly once we've identified a particular gene mutation in a family, then those unaffected relatives can potentially undergo genetic testing if they wish. And, we work with families in the pre-fALS study, and as you know, within a family, everyone has different personalities, everyone has different viewpoints, and this is something that we definitely have to take into consideration. And you can see here, some of the different things that people are thinking about, when they're considering the possibility of doing genetic testing. There are going to be those people who think, oh, of course, why would you not want this information? Why would you not want to, potentially have this knowledge to share with other family members and pass down for the future? But other people, that's a little more concerning to them. They're not sure. Do they want that definitive answer? yet are not. And people come at this with different backgrounds, different experiences. And again, very, very important to think about within a family. So, really for someone in that situation, where we are is learning, does someone really want to learn their true risk? Are they at risk for developing ALS or not based on their genetic makeup? And how do they feel about knowing for certain versus that uncertainty that most people have been living with for some time, knowing what their family history is? And these are a lot of the considerations that people have shared with me and that I know people are thinking about, and that we encourage people to think about as they're kind of pondering, is genetic testing the right step for them to take? Things like, how is it going to impact themselves? How is it going to impact their family? How would they feel if they did turn out to be positive for a gene mutation? I mean, one thing that's important to realize is that if someone's having genetic testing and they come back for genetic test results, we're going to say yes or no. And once that information is out there, we can't retract it. So we really want people to take some time and think about how they feel. I know no one can predict 100%, how they might react to a given situation, but at least take some time to think about the potential implications and feel as though they're, as potentially ready as possible to learn that information. Other things, how would it change someone's life or not? And in some cases, people feel like it would alter certain things about how they, decisions that they make in their life. Other people feel like that's not necessarily the case for them. Who would you tell if you turned out to be positive or negative? Again, a lot of people have big families. People are very active in the community. Some people are sharing information. Some people may not want to share that information, and that's important. And there's also a phenomenon called survivor guilt. And survivor guilt is the idea that, someone tests negative for a gene mutation that's in their family, and while that's generally considered on a personal level to be a positive thing, there may be some negative feelings associated with that, because they may have the feeling of, okay, why was I spared? Well, my brother, sister, whoever else is having to deal with this particular situation. And that is very real and very impactful, and something that people aren't always expecting. Additional considerations, and all of these are things that we go over in the course of a pre-test genetic counseling session. And sometimes these come up, sometimes, again, they're more forceful people in the family that may be driving the situation. We want to make sure that that particular individual is really the one making the decision and being comfortable with that information that they're going to receive. It is possible to participate in genetic testing under the auspices of a research study, and different research studies are set up differently in terms of what's the likelihood of getting results back, what's the timetable on which you're likely to get results back. But it's also possible to do testing for a lot of these genes now commercially as well. And that's something to think about. Commercially means it's available by for-profit laboratories out there, so you pay the fee to have the test done. Will testing impact my insurance? And we'll touch on that a little bit more in just a minute. And what about testing for children? So typically, we think of ALS as more of an adult onset disease, and generally speaking in the genetics community, we do not advocate testing minor age children for conditions that have more of an adult onset, especially when there's no knowledge of any intervention that we could do during childhood that would impact their ultimate disease risk. So we generally, if someone were tested and found out to be positive, if they had minor age children, we generally would encourage waiting and sharing that information with those children as they got older, so that they can make their own determination about whether or not they felt comfortable pursuing genetic testing. So some of the reasons people have shared with us that they actually choose to do genetic testing, of course, probably first and foremost to have information to share with their families, especially with their children, and potentially, if they test negative, to alleviate anxiety in their kids. And sometimes for themselves, really to alleviate some of the anxiety and the ambiguity that they've been living with, kind of wondering which side of the fence they fall on. You know, are they truly at risk or not? And to make appropriate lifestyle decisions based on that information. Some reasons people have shared that result in them choosing not to have testing or choosing not to have testing at a particular time. And, you know, this is one of those things, genetic testing is sort of out there, so it's not a limited time offer. If it's something that someone's considering today and they decide for whatever reason that it's not the appropriate time for them, that doesn't mean the door is definitely closed forever. It means, you know, if a year, 2, 5, 10 years down the road, they feel like this is something that they want to pursue, that's still going to be available to them. So it is something that's not a one-time decision. But, you know, some people are concerned about how they would react themselves, concerned that they would regret learning that information, and some of that fear of survivor guilt. You know, I have seen people who felt very strongly over the course of their life and their family experience that they were going to be positive, that they were going to have a gene mutation, and actually finding out that they did not was very significant for them in terms of, you know, they're now having to sort of reassess what they thought their life plan was going to be. And, you know, again, all of those are important things to think about, and people are going to react in different ways. So I wanted to take a minute here to talk about insurance issues, because this is something that often comes up in terms of questions that I get from individuals being tested, and also questions that they have regarding potential insurance for their children down the road. There is a federal law called GINA, the Genetic Information Non-Discrimination Act. It was actually signed in 2008, went it fully into effect in the fall of 2009. And what this GINA law does, and because it's a federal law, it sets the minimum standard of what has to be done. Some states have their own independent laws related to the use of genetic information in the insurance industry, and if those laws are more broad even than GINA, then they supersede GINA, but GINA sets the minimum standard. And what GINA does is it prohibits discrimination by health insurers and employers based on genetic information. So what do they consider to be genetic information? That's your genetic test results and relatives' genetic test results going back for at least 4 generations, so going back to and including great-great-grandparents. And information about participation in research studies that involve genetic testing and genetic counseling is also protected under GINA. And most research studies also have additional protections to guarantee patient privacy as well. So what sort of health insurance protections do we have under GINA? First of all, this law does include people who are insured both through group and individual insurance policies, also Medicare supplemental policies. And it says that insurers cannot use genetic information to set eligibility, premium, or contribution amounts. Now, you know, all laws unfortunately leave a few people out. This does not cover members of the U.S. Armed Services, service members covered under TRICARE, people covered through the VA or through the Indian Health Service. So what GINA does not do, and this is important as well, it does not restrict providers from offering testing or providing information. It does not require insurance companies to fund genetic testing or particular treatments, although a lot of companies are paying quite well for most types of genetic testing if there's a specific reason based on family history. This law was particularly designed for people having pre-symptomatic testing. So it really does not impact people who already have a diagnosis. But there are other laws in place, the Health Insurance Portability and Accountability Act, as well as the Affordable Care Act, that do offer some protections for those individuals in terms of the issue of pre-existing conditions and changing insurance companies and things of that nature. This law also does not apply to things like life, disability, and long-term care insurance. And I don't know that many states individually have any policies related to restricting the use of genetic information by those industries. So that's something to consider, and especially when I'm talking with young people about doing genetic testing, you know, even though there's not a lot to suggest that people are having issues with getting these types of insurance policies, but if someone's young, they maybe haven't thought about life insurance yet, things of that nature, you know, I do suggest that it's something they may want to consider prior to undergoing genetic testing, just to sort of err on the side of caution. So how to access genetic testing? Again, this can be done currently through research studies, and there are commercial laboratories out there that offer genetic testing for various ALS-related genes. Now, it's very important if someone's thinking about doing commercial testing that they do try to engage the help of someone who is familiar with this field, because there are lots of different things out there. Labs are offering lots of different alternatives. Some are offering big panel tests where you can get, you know, several genes done at once, and those tend to be quite expensive, and expensive on the order of probably several $1,000s. You can do individual gene tests. If there's a particular mutation that's known in the family that someone's at risk for, then if you have a savvy person filling out your order forms, you can order a test for that specific gene mutation, because we expect that if we've identified a particular gene mutation in a family, everybody else in that family who's at risk is going to have that same gene mutation. So we don't have to look at the whole gene anymore. We can just look at that one particular spot. And at that point, we're talking about a lot less in terms of cost. Testing for a single gene mutation typically is on the order of about $350 to $450, maybe $500, depending on the individual laboratory, whereas testing for an individual gene is on the order of, you know, $1,000 to $2,000 plus, depending on the size of the gene. So how can someone access genetic counseling services? There are a couple of good places where individuals can go to try to find a genetic provider close to them. And again, because of all of the different options in terms of testing, all of the different issues that we want people to carefully consider when they're thinking about undergoing genetic testing, we really do encourage them to try to find someone who's knowledgeable in this area. You know, again, some of the commercial laboratories may be out there sharing their information with you know, independent care providers, primary care physicians, and saying, oh, you can just order these tests, but it really is helpful to have someone who actually knows the subtle nuances of how this testing is done and what the potential implications are to navigate the process. So NSGC is the National Society of Genetic Counselors, and they have a Find a Counselor option on their website where you can type in a zip code and look for genetic providers close to you. There's also a company out there called Inform DNA that employs certified genetic counselors, and they actually do counseling primarily by phone, so people who are not in a geographic area easily served by a genetic counselor can access those same services and help get testing coordinated through someone who does have that knowledge. And just to share with you, this is the National Society of Genetic Counselors website. When you go directly to their homepage, you can see right across the top, there's the bar Find a Genetic Counselor, and you can easily get to this page where you can put your zip code in and see what's close to you. And then this is Inform DNA. They do lots of different things. This is their adult genetics page. They do not specifically list ALS there, but I have spoken with their counselor. They do have a counselor who does specialize in adult onset diseases, and they are versed in this, and it is something that they can help people with. And then just as one additional reference point, this is something that was put out a few years ago by the Surgeon General's office. This is a website that can actually help you draw a family tree or help you track certain things through your family. And it's called My Family Health Portrait. And it's undergone some revisions over the years and is now quite user-friendly and is something that individuals can use if they want that sort of tool. So I think we've got a panel coming up next, and I just want to thank, again, the ALS Association for inviting me today. All of the wonderful families that I have had the privilege to work with over the last several years as part of the fALS study, and also Dr. Benatar for coming to us with the, with the question of, do you have a genetic counselor who can work with this? Because it's really been a tremendous opportunity for me, and I've really enjoyed it. Thank you. And I also want to add, I did print out some copies of a really nice summary of the GINA law, if anybody is interested. This is from a joint publication actually by a breast cancer advocacy group and the National Society of Genetic Counselors, but it gives a very good summary of what that law does.
Thank you so much for this excellent presentation. And now we're a little over time, but I think we've got 20 minutes for a good session of Q&A, and I'd ask all the speakers to join us here. Is there a mic for the floor? Oh, here it is. Oh, Kim, thank you, Kim. If you don't mind just to field, and I'll try and look out for where the questions are. So please feel free to ask any questions of any of the panelists that came to mind as you were hearing the presentations, or anything that's outside of what was presented would be welcome. Thanks.
My name is Mary Johnson, and this is my first summit. I've lost 9 members of my family so far. The most recent, on March 13th, my very young 34-year-old niece. And one question that I have for the panel is, we were told that if you have, and we have the SOD1 gene, that we would be related to others who have the SOD1 gene. Is that true or not true? Or
So that's a very interesting question. It depends on what mutation. So if you look at the SOD1 gene, it's reported that there's about 140 or so mutations within the SOD1 gene. So it's made up of all of those different letters that Michael talked about. So any of those letters could be mutated, could be changed. Individuals who carry the same mutation are potentially related to each other. So you may not know, you may not be able to trace back your own family lineage, but from a genetics perspective, we could look at that DNA and determine whether whether those two individuals are distantly related to each other.
Great.
Welcome.
Hi, my name is Dawn Holcomb, and we've lost 4 family members on my husband's side. He passed away in January, and we also have a sister who currently has it, and she's in the end stages. It seems like almost all of them, I feel like they got triggered after some environmental exposures, because within maybe 3 to 4 months after they did have an exposure, and 2 of them were tested and had lead, and my husband had like 6 solvents out of 10 that were really high in his blood levels. So I would like to know if there's more emphasis being put on, because I feel like I had to argue with so many doctors and neurologists until we finally got to Henry Ford Hospital in Detroit, because they don't know what to do. So you have a group of neurologists that know what to do with ALS patients, but they're making guesses. And they'll say, here's the EMG, you're twitching, you have motor degeneration, yeah, you have ALS, but they don't go a step further. So they really need to be educated, because it's so stressful. I have a strong medical background and have done research in ALS in my master's degree program, and I can be a good advocate for them, but there's so many people that don't have that, and our doctors need to know what to do. So I just want to know what promotion there is to help that.
So I think you raised several important issues. I mean, I think many people working in the field recognize that it must be the case that there's some environmental exposures that are important, but really getting a handle on what those are and how relevant they are across different people with the disease has been incredibly difficult to pin down. I think one of the things that came out in Dr. Trainer's talk is that one of the reasons we've made the sort of progress we have on the genetic side is because of the genetic and genomic technology has advanced to the stage where it's a tractable problem to figure out the genetics. Hence his optimism that this is something that can be solved genetically, at least into the genetic component, within the next 5 years. Figuring out the environmental stuff is so much more complicated, and we really lack many of the tools. I think it's an area that still needs to evolve and is evolving. And I know, for example, that ALSA, through their science program, is supporting efforts to better figure out what some of those environmental exposures are. And I think part of the confusion is that the truth is we don't really know. And people have beliefs, they have ideas, there's sort of red flags that come up, there's sort of smoke, but hard evidence that this is the case, this exposure is relevant, has been incredibly difficult to come by. So I think part of it is that it's not clear. Lucy, I don't know whether you want to add to that.
I can make a few comments. I think you touched on it. I mean, we certainly are trying to help support environmental studies. They firstly are extremely expensive. So we have formed partnerships in the past with the National Institute of Environmental Health Services, NIEHS, and we are this year currently going to fund another small study. But again, it is a numbers thing, because it's not enough to feel that there's a suspicion of something, because sometimes it could be a coincidental situation. And for example, brain trauma has arisen through many various venues. We have a large Italian study going on that is being done, including soccer players and others, and trauma is one of the questionnaires. But it is incredibly difficult to really pinpoint. And the other challenge is it's very likely a combination of things. It's probably not just the one. And how much earlier was that exposure as opposed to, was it multiple exposures? Some things like lead could be measured perhaps, although even in discussions with others, more recent exposures are difficult to capture in just a blood sample. You need much earlier samples, so bone. And so I think the realm of the studies are so complicated. We do have a small cohort of really good ALS epidemiologists working with us globally, but that's fairly limited. So it is the challenge.
It's a tremendously important issue to figure out.
And in terms of education, the difficult thing is if we as scientists and colleagues haven't quite figured it out, to get a good solid message then to people who are even less versed with this is very difficult too. Although I think that doesn't mean we shouldn't be doing it. If it's difficult, we have to try and find ways to communicate.
And another point to make, and perhaps modesty on Lucy's part precludes her from doing this, but ALSA goes out and funds clinics for ALS, and part of that is educating the neurologists so that they know what's available in the community, resources that are available in the community, and so forth and so on. So I think that ALSA really is making efforts and big strides in education of physicians as well.
Hi, this is a question that actually regards the sharing of the collaboration between researchers. And in the particular instance, I'm referring to the wet repository, the banking. I think Dr. Benatar mentioned it. I'm George Bauer. My wife, Julia, has ALS. And so far now, we have sent blood from 2 siblings in that family and 1 nephew. That went to Dr. Sadiq at Northwestern. And now 2 of those individuals have passed away. And we're worried that those samples that perhaps are valuable may just be sitting there. Are those samples shared across researchers? And how do they get access to things like that? I believe those blood also was sent to Dr. Benatar's office.
So this is a great question, and I think a tremendously important one. And I can't sort of speak on behalf of what other investigators do. Any samples that we receive, we try to make broadly available. We share I guess we're equal opportunists, as it were, with obviously your permission, and this is an important issue. When samples are collected, we try to get your permission, when I say an individual's permission, to share those samples in a de-identified fashion with other investigators. So there's a mixed message there, right? Because we want to guarantee your confidentiality, so we want to share things in a de-identified manner. So that means when the samples are shared, that other investigator can't directly trace them back to you. That information's got to come back to the investigator who collected it, who then has your information to be able to share it. So we want to share as scientists. I think we do try as much as we can. We have to navigate very carefully some of the sort of personal, the confidentiality issues. What we try to do is to share our samples through national consortia that other scientists can access those samples for research, but always in a de-identified fashion. And I think Dr. Traynor can speak as well to the value of having de-identified samples, but also the challenges. When you find something and want more information, you can't get it because the sample's been de-identified. And so I think there are issues that as a community we haven't fully worked out, but it's part of the barrier is making sure that we're observing people's confidentiality.
Yeah, I would absolutely agree with Michael. I think, we all want to do research, but at the same time, we have to recognize that there are, I think dangers might be the wrong word, but potential pitfalls with respect to this, because you have your information. This is private to you. Your diagnosis is private to you. You don't want that shared. You may, but not everybody may want that shared with the entire world. The only other thing I would add is that that what you're talking about, believe me, there's growing recognition over the last decade or so that this is a serious problem, not just within the ALS field, but within other disciplines as well. And there is a repository that was set up by NINDS, which is a branch of the National Institutes of Health, with funding from the ALS Association and MDA. It's called the Coriell Repository. And basically, this was a fairly large effort on the part of all neurologists around the United States, collecting samples and depositing them in this repository. Again, de-identified, as Michael mentioned. The advantage behind this is it is, you ring up, you talk to the company, you get your samples. There's no barrier to that. You don't have to go through scientific committees or things like that. So that repository is excellent, and believe me, an awful lot of the studies that I've done have used that as the central collection. Can we do better? Yes. Are we trying to do better? Yes. There are other efforts underway to expand that Coriell repository and to make other repositories as well.
And I would just say, from a funding perspective, the ALS Association in most cases puts clauses into, and the agreement is with the researcher, that they should be made available. That's true of our mouse models and other things, but particularly also of this, but de-identified. But we certainly encourage through our funding priorities to only fund on condition that it's used collaboratively.
Also just add that I think because of that, there are these issues of sharing confidential information, that different investigators are often approaching problems in different ways. And I think there's value in sort of donating samples on multiple occasions and to different laboratories, because I think different people are sort of using their valuable research resources in different ways and might come to different answers. And so I think as much as we try to share, I think what you can do as a community is volunteer and participate in as many different studies, because different people may come to an answer in different ways, and I think there's value in that too. And that's not to say we don't need to do a better job of sharing, we do, but I think it's another short-term approach to helping to get as much information that you can.
I absolutely concur, and so there's no compromise. If you've been in one study, there's no compromise by going into multiple studies, and that's true of the registry, the National Registry, the fALS registry, Coriell. Really, it's a blood sample, so fortunately that's not a difficult thing to contribute multiple times, and really I think it's important. We will often on our association website help investigators and say, could you please contribute and be involved in the study. Never feel that that's going to compromise your other involvement in studies. Sometimes we're explicit about that, but you should really, if we put out a notice and you can contribute and would like to, you should feel very comfortable to do so.
My question is really around, I'm Shauna Hackett. I lost my husband in February, and we had two other members prior to that, his mother and also his grandfather that we know of. The family history is a little sketchy, but it is the SOD one, so maybe you're related. But really around the trauma issue, because both with my husband and my mother-in-law, almost a year to the day of their diagnosis, both of them suffered a trauma. My concern comes in as I have a 16-year-old who had a horrific leg injury from a soccer injury. Obviously, we haven't had him tested, and I'm not looking to go that direction, but that's definitely in the back of my mind, and I've heard a lot of, I haven't been able to find a lot of research around the trauma, but they both seem to correlate with their onset of their diagnosis. So if you can just expand a little bit on that.
So I think one of the challenges to figuring out any environmental exposure, including something like trauma, is that we tend to remember things selectively, in the sense that when we have a problem, we have a medical diagnosis, we sort of look back and say, what could it be that caused this? And that's commonly done because we're trying to sort of figure it out and understand it. And so we look back and we think, was it this, was it that, was it the other? But of course, the flip side of that is the people who've not developed disease, who are not sort of looking back and asking, did I have this trauma? Did I have this exposure? Did I have something else? And so I think the relationship between trauma and the onset of disease remains unclear. I will say that people have asked this question before in multiple sclerosis, and the MS community went through this and eventually sort of came to the conclusion this was not a risk factor for precipitating MS. So that's not to say it's not something that we need to think about and research, but I think it's something that remains uncertain.
Yeah, and again, very difficult to do because, in many cases trauma is not associated, in some cases it is, and whether there's a connection. I mean, there are studies ongoing in terms of traumatic injury and looking for markers and changes, but it's very difficult to correlate, and I think we have to be very cautious as a scientific and clinical community to not correlate the wrong things, because again, that would actually derail the direction of research in a very unsatisfactory way. So I think we have to be careful of assuming incorrect associations.
Yes, my name is Mark Manchester, and I was diagnosed with sporadic ALS about a year ago and was fortunate enough to be accepted into the NPO1 clinical trial and was quite surprised that people with familiar ALS were not accepted into the trial. And, we've had some positive results with the trial. And looking at Dr. Benatar's slides, it's like there is once the onset occurs, there is some, the symptoms and the progression is very similar. So my question is the research for sporadic and familiar going in different directions? Or is it, that's a question.
So I'll quickly answer that and leave it to the clinicians to really. So I'm not aware that in any trials there's that's a criteria to eliminate. Certainly for a particular familial ALS trial, there's a requirement. Most cases, the restriction is more on the length of time that someone has had the disease. But certainly, I think that everyone commented that clinically it's very similar, in fact. So I think it's best of you to comment why there would be a discrimination, and I'm not sure in this case why for that trial it was the case.
So one possibility is that although we emphasize the similarity and the overlap and sort of the spectrum between familial and sporadic ALS, it's also the case that ALS may be more than one disease, or at least may have more than one cause. So, for example, SOD1 mutations can be a cause of ALS. Mutations in C9orf72 can be a cause of ALS. So those are definitely two different causes producing the same disease. And the underlying biology, the mechanisms, whilst they may share a final common pathway of the motor neurons being sick, there may be more sort of upstream events that are different. And so if treatments are being developed where they target mechanisms that investigators might think are relevant to a particular kind of biology, they may want to restrict the trial to focus on that specific population. And so I can't speak to this particular trial, but there may be powerful biological reasons why you want to focus on a specific population of people. And that may be one of the future directions that ALS trials will go. That as we know more about the genetics, it may be appropriate to say, if you have this gene, you should be perhaps in this trial. If you have this gene, in that trial. If you don't have a gene, perhaps in a different trial. We may not yet have reached that, but that may be one of the future directions, because one size may not fit all. I don't know whether that's the case, but that's a possibility and something we at least need to consider. Do you want to add to that,
Yeah,
Brian?
no, I think you voiced it absolutely perfectly. I mean, basically, one of the perhaps, and this idea has been raised, that one of the reasons why clinical trials in ALS have not really been terribly successful, as successful as we would like them to have been, is because the disease is very heterogeneous. So say you had 10% of the population where a drug actually had an effect, you'd miss it because it was swamped out by that other 90% where it wasn't. So the idea is, can you enrich your population that you're starting off with, your cohort that you're starting off with, in order to try and see that signal to start with? And, I think that that may have been the genesis behind them restricting it to just sporadic disease. But I agree with Michael that as we move forward and as we understand more and more about the genetics, probably we will end up doing clinical trials that are honed in on one particular type of ALS.
I would just add, and Michael's got the best example. I remember years ago now, it has been quite a while, when he had come up with the idea of why don't we test specifically SOD patients and which compounds should we be thinking about. We did dialogue back and forth, and the reason that arimoclomol became the one to choose is that mechanistically we think it works through a folding and helping the inappropriate folding. And so that was very specifically designed because we might have that problem with now that new genes have been identified, TDP43 and others that have folding problems. But at that time, it wasn't clear that in sporadic disease, folding problem was a general problem. So that's exactly why this trial with arimoclomol is going on. So it's sort of the reverse of what you're indicating in that particular trial.
And then we'll come back and sit on these tables. So we want to make sure that everyone has time to eat. But I think we have 2 more questions then.
She talked about are we related? 3 years ago, we started charting out our family, and we only had a small portion of this done. Last year, we connected and we met Steve. So we have 3 generations here today across 13 generations of 300-400 years worth of family for us. Steve is the Gilson line. Michelle Farr is the Farr line. And I'm part of the Underwood, my daughter Kristen. So across to what we've done in the last 3 years, we have identified 68 patients within our 3 families. The pink is the carriers and the orange are the negatives. So are we related? Yeah, it does prove that we are related through the genetics that we've done. Michelle has done a great job creating a blog online for the familiar families. If you're not familiar with us, it's called Making Connections. So you might want to be able to check it out.
So we have a final question here.
My question was regarding the clinical trials. There are restrictions as far as time from diagnosis to getting into some of the trials. And since, especially with familial, you tend to get diagnosed on the earlier side if you're aware that it runs in your family, is there any chance that they're going to base it more on your clinical assessment versus your time from diagnosis? Because I'm over a year from diagnosis, but still clinically doing very well.
So I think it's very individual trial specific. I mean, different trials are designed in different ways to enroll a different population. And there's always a challenge in designing a trial that you want to be as broadly inclusive as you can, but also sufficiently focused that you can get a meaningful answer to the biological question. And so I think if you're looking at an individual trial, discussing the specifics of the inclusion and exclusion criteria with that investigator and understanding why that is, I think is important. But I think the reasons for specific criteria will differ across trials. It might be that the biology is relevant to this particular treatment and this particular gene. It might be that the drug is expected to work earlier rather than later in the course of the disease. There are all sorts of potential reasons, and they could differ across trials.
So I think with that we are running a little over time, but I firstly want to thank the panelists for tremendous presentations and keeping to time and giving us very, very valuable information. I think it's been tremendous, particularly to all of you for your good questions and your patience in listening to some very complicated and complex studies. I'm very pleased we had this opportunity. The ALS Association's research program is committed to continuing to fund genetics research and will continue to look for your participation. And so we're really pleased that you were able to participate in this session. So thank you again to the speakers.